In this video, I'd like to show how to navigate the main data display in Toxygates. So the first step to doing that is to define sample groups here on the sample groups definition screen, which I have discussed before in a previous video. So currently I have two groups defined. One of them is with acetaminophen, middle dose 24 hours, and the other is with cisplatin, middle dose also 24 hours. Once you have defined at least one sample group, you can go to the View Data screen either by clicking here on the View Data tab or by clicking the Next View Data button down here. So I go to the View Data screen, and this shows you the gene expression in the sample groups you defined for every gene in the system. So in this table, every row is a gene, for example, UDP glucuronidase transferase 2 family, phosphoglycerate dehydrogenase, and so on, with a gene symbol, a probe title, and a probe ID. The green column corresponds to one of the groups you defined as the acetaminophen, and the colors are automatically generated for you. And the red column corresponds to the other group, cisplatin. And currently you see only 50 rows out of 31,000, which means you can use these paging controls here to move forward or back. And the left and right arrows move in small steps, and the double arrow moves in bigger steps. So currently the data is sorted by the log2 fold value of acetaminophen, middle 24 hours. So out of those 31,000 rows, you will see the largest values at the top here, and as you page forward, you will see the values getting smaller. And at the very end would be the smallest values. You can change the sort order by clicking the column, clicking on the area where it says log2 fold here, next to the arrow. And this is reverse sorting, so now you see the smallest log2 fold value first. And this would mean that there is almost 32 times less of the gene expression in the treated sample compared with the control sample. Since it's log2 fold, it would be 2 to the power of 5, which is 32. If you click on the red column in this area here, you can sort by that column. And now the order of the genes has changed again. Now you see only the genes that have the highest upregulation in treated versus control for cisplatin. And clicking again places the greatest downregulation at the top, the most downregulated gene. You can only sort by one column at a time. However, you can set filters on columns. So for example, if I click here, this little icon is the filter icon. I can set a filter such as 2, and the filter type can be one of these four. In this case, it's absolute value greater than, which means the log2 fold value should be at least 2, absolute value. Now you see only the ones that are either smaller than 2 or greater than 2, and that limits the total number of rows to 85. So now you're looking at a much smaller subset of the total space. At the same time, we can also filter a cisplatin. I set the filter to 1. And now you're down to only 7 rows, only 7 genes, which were significantly changed by both acetaminophen and cisplatin. You see that this icon also becomes blue to indicate that the filter is active. To clear the filter, you can click on this blue icon and then click Clear Filter. And that resets the filter, and you go back to a greater number of rows, 210. And if I reset this filter as well, you're back to the full display of 31,000 rows. If you check this box here with p-value columns, you can see p-values as well in the case of log2 fold values. This will be the t-test p-value, student's t-test of treated against control samples in that column. This is quite a simple significance test, but nevertheless it can give us some idea of the significance of some genes. And as I just did, you can also sort by the p-values like any other value, and you can also filter and say that p-value should be, for example, less than 0.01. And in this case, the filter mechanism works exactly the same as for gene expression values. So I'm going to clear that filter again.